Hello friends, welcome to BMH Learning. This video is about green fluorescent protein tagging. GFP protein was first isolated from the jellyfish Equioria victoria. The protein is consisted of 238 amino acid residues. Now, the property of this molecule which attracts the interest of a biologist is that it fluoresces green when exposed to UV light. So it gives green light. The GFP gene was successfully inserted into E. coli bacteria in 1994. And for the discovery and development of GFPs, in 2008, the Nobel Prize in Chemistry was jointly awarded to these scientists. Because the GFP fluoresces when exposed to UV, they are used as a reporter of gene expression in molecular biology. So, for this purpose, proteins are tagged with GFP through certain genetic modifications. And this makes the protein to be observed through fluorescence microscopy. Now let's talk about the basic idea of GFP generation. We know fluorescence microscopy uses light in order to study specimens. So we require a fluorescent chemical, referred as a fluorophore, which can absorb the light of a particular wavelength. And once it absorbs that light, then it emits light of longer wavelength. So GFP tagging is nothing but a certain way of preparing a fluorescence microscopy sample by using the GFP as a fluorescent protein reporter. So basically, this is done through attachment of the GFP with the target protein. Now, the GFP can be attached either at the N or the C terminus of the amino acid chain. Now, by using GFP tagging in fluorescence microscopy makes it possible that the specific proteins within living cells or proteins in which we are interested can be visualized and studied. Use of mutations in GFPs. Now, there are many types of GFPs and some of these are not useful for us because they observe multiple wavelengths of light which makes them unsuitable for fluorescence microscopy. And also some GFPs shows reduced brightness and photostability. So to make these GFPs suitable for use, we need to enhance them. And that's done by causing mutations in them. So, one way of developing mutant GFPs is to allow colonies of bacteria with GFP expression to grow and wait for mutant variants of GFP to develop with time. These mutations increase brightness and photostability. Apart from this, the mutations also shift the excitation peak of the GFPs to match the spectral characteristics of the commonly used light filter in fluorescence microscopy. Further, mutations also increase the fluorescence yield. This means there are now enough fluorescent proteins to cover nearly the whole visible spectrum. This was all. Thanks for watching.